Yeah, that was just incredible, and really excited to be here. And tonight, I want to take you all on a journey to an alien environment. This environment just happens to be on our planet, but we know less about it than we know about the surface of the moon or Mars. I'm talking, of course, about our oceans, which make up an incredible 90% of the habitable space on our planet. Despite this being one of the largest ecosystems, it's powered by single cells that are so small we can't see them with our naked eyes. For example, single cell phytoplankton, which are the equivalents of plants in the ocean, fix 50% of the global carbon every year due to photosynthesis. Every other breath that you take is thanks to oxygen that directly comes from these phytoplankton. When these cells die, they clump together and sink in what's very poetically and aptly known as marine snow particles. If you've ever seen an underwater video, it really does snow under the ocean. These marine snow particles transport vast quantities of carbon from the upper sunlit ocean to deeper waters. This is called the biological carbon pump, and it's so powerful and so remarkable that in a given year, it transports as much carbon to the deeper ocean as we emit in a given year. And a fraction of that actually gets buried for very long-term sequestration. In a very real way, the oceans play a direct role in regulating our climate, yet are powered by these processes that we can hardly see. How do we bridge those scales? How do we bridge the scales from the depths of the ocean to the scales where these processes are happening? This was a problem that flummoxed me for a really long time as a PhD student. And the aha moment came when I least expected it. My advisor, Manu Prakash, and I were sitting in his office planning a field trip to Madagascar. And we were really interested in studying parasitic organisms and their distribution in the water column. And we realized how impractical and hard it is to pack a microscope that's meters in size. What we realized is there's a simple solution to this problem. It hit us that we could take a tube of fluid and actually just bend it back on itself to create a loop or a treadmill. Using our knowledge of fluid physics and microscopy, we could build a light microscope at a particular part of this tube and essentially let a cell or a particle move infinite directions in the vertical direction and compensate for that by rotating the tube in the opposite direction. So in some sense, we had access to infinite vertical scales for free in the lab. And this led to this microscope that we call Gravity Machine. As soon as we had an idea for this microscope, though, I was deeply uncomfortable because I realized I had never built a microscope in my life, let alone one that could traverse like infinite vertical scales. And this was really my introduction to the power of interdisciplinary science because I had to move really freely between optics, mechanical engineering, software development, and fluid mechanics to actually make this microscope a reality with an interdisciplinary team. And I'm really happy to say that within 2 years, we actually had a working microscope in the lab. And the first thing we did was to put that in a suitcase and take it all across the world to where there are the most interesting marine samples. These were marine stations in California, Puerto Rico, and even eventually sailing it on a research vessel in the North Pacific. The kind of things we could see with this microscope were truly mind-blowing because it was really like it had opened up a window into the microscale and the ocean. For instance, we could float alongside a diatom cell and watch it change its mind from floating to sinking. We could watch a baby starfish larva and swim alongside and watch its behavior in minute detail in a way that was not possible before. And lastly, we could watch these marine snow particles take birth and basically sink alongside them as they transformed and sank through the water column from the comfort of the lab. All these experiences showed me the power of this frame shift from thinking about the ocean at a bulk or population scale to really paying attention to stuff at the micro scale. And thanks to this opportunity through a Schmidt Science Fellows pivot, I was actually able to make this a reality by pivoting into cellular and molecular scale biophysics at the Fletcher Lab at UC Berkeley. Here, I'm actually developing tools to actually understand marine snow and its components at the cellular and molecular scales. For instance, by inventing tools that allow us to measure the stickiness of cells that make up marine snow, as well as molecular glues that hold these particles together. The goal of this work is to one day predict the properties of marine snow given knowledge of ecosystem components in a given location, which you could get from direct sampling or from satellite imagery of plankton. I want to switch now to talking about the oceans not just as a source of worry about how they're going to help us combat climate change, but for another big reason that I study them, which is a very strong sense of wonder. Every drop of seawater that I've collected has shown me that it contains untold biological treasures, especially at the micro scale. This was made very real to me recently when I collected some samples from the San Francisco Bay, and I found in there a cell that did something absolutely remarkable. The cell I found was a choanoflagellate, a type of protist. Protists are enormously important in marine ecosystems because they're single cells, but despite being single cells, they're so numerous that they equal all marine animals in terms of their biomass. The protist I found, this choanoflagellate, was remarkable because it could biomineralize silica and weave itself this most exquisite basket around itself that was completely remarkable. As a mechanical engineer seeing this for the first time, I literally fell out of my chair because I knew just how hard and what engineering and physics challenges this cell was solving in order to make this structure possible. Every day when I study these cells, I'm amazed and fascinated by what new molecular secrets they're going to teach me. And in many ways, I feel like the pioneers of flight probably felt when they saw birds flying effortlessly for the first time. This cell is a real North Star of possibility for how we can one day build these structures ourselves. To end, the oceans are really what make life on our planet possible. And despite this, they're organized at these scales that are very inaccessible and small. And at these scales, they also hold untold biological treasures. I worry, though, that these treasures are changing and disappearing as we speak. This makes everything I do be imbued with a real sense of urgency. It's only by studying planetary health with the same intensity and urgency that we study human health can we really seek to understand how deeply intertwined the two are.